G'day, good morning, good evening. Thank you for listening. Today's going to be blooming excellent. Oh, the next few weeks are going to be excellent. I'm all excited. I had to calm down before giving this intro. Anyway, this show is about agricultural, horticultural, and gardening things. It's focusing on plant science, soil science, and microbiology, and the information is fact-checked and can be relied on for educational and scientific purposes. Any of the papers or websites that have been discussed will be put in the description below. And if you're rewatching this back, fast forward until you see the screen change, or some slides pop up, because that means that's pretty much when the show starts, because who knows what's going to happen between now and then. But pretty much I'll give it till 2 minutes, so another 60 odd seconds, and then I'll start. And that just gives it a bit more time for people to get into chat, because there is a delay here, and I like to interact with chat people if there's questions or discussions and stuff like that. So I won't talk about microbes yet. 50 more seconds. Wait. It's hard for me to wait. 40 now. I've been looking forward to today for a while. It's going to be good. 30 seconds. $20 seconds to go. Getting lower. 15. $10 seconds. Here we go. 5 seconds. Get ready, get ready, get ready. Yay, it's microbes in agriculture. It's going to be so blooming cool. This is a course that I just did. Let's share the screen so we can get some interaction. Doesn't look like there's any chat at the moment. So I want that screen. Yep. So this is the course that I did. Microbial biotechnology. I got 85%. The exam, got it back a few days, about a week ago. So I'm going to go through next week or so, depends how long this goes for. This was the course here. So I'm going to go through all those slides in PDF form. Yeah, I'm not going to go through because I can stick to our topic a lot easier. You're going, what's he on about? That's right, just start. These are the topics for today is section 1, section 2. I don't think I'll get to section 3. But so I'm going to extensively go through these for once, instead of just skimming through and getting you guys and girls to pause the video if you want. I'm just going to go through them pretty good, and I'll stop it regularly and see if there's any questions. So that's a bit small, isn't it? Can't really read those words. Is that better? A smidgen better. And you can't get the whole slide in. Righto. Well, microbes in agriculture. This doesn't really start on this is already into week 7. So it's not really an introduction into it. We're already into it, and we already know about how microbes work, their functions, what they do, what they can't do, etc., how to culture them, all that sort of stuff. And now we're up to this bit. So this more specifically goes into detail about like the soil microbiome, for instance, and right into detail about, this course was written last year. So it's up with the latest agricultural techniques with how to manipulate and make microbes work for you. It's just blooming excellent. And as you know, as humans, having microbes in our bodies, 90% of our body's cells are microbial. So when we get anything that interrupts them, it will have adverse effects on our body as well. Well, the plant's the same. They're eukaryotic cells, and they have similar effects through microbes. So you can see here on the left, unhealthy guts, you know about the brain, the gut-brain axis pathway, how they're just looking into that and they're finding it's very closely related. Fat people have different microbes in their guts to skinny people, for instance, and that's all I'm going to go into about the human stuff. So the left side here, it shows there's all sorts of pain and problems, and on the right side, it say the well-being's quite good, and on the right might be because the gut microflora is just thriving, and it's symbiotic. It hasn't been pumped with antibiotics and anti-this and killed off with bad things like or glyphosate, for instance, because that affects the shikimate pathway in microbes, which is affects their metabolism. So things like that. So that's what the effects are in humans, and it's the same thing in plants. So we're going to find out how it actually, and why, how you can fix it and make it all thrive. So here on the right, I'll read off the slide. So the healthy ecosystems on the left and the unhealthy is on the right, and you can see the big difference. Because on the right, everything's gone. It's had a recent fire. The fire might have been so intense that it's gone in and affected the O horizon and A horizon in the soil, which is most of the microbes. So therefore, to try and get this back, bounce this back, you're on struggle street. So there might be some pathogens that are going to thrive now. The invasive weeds that are more sturdy, they're going to thrive. So this is all to do with microbial interactions. So the accumulation of undecomposed litter, reduced soil for functionality. So on the left, the healthy stuff, as the soil organic matter stabilised via the soil fauna breakdown with microbial decomposition. So everything, saprophytes, everything's working within it should be. Has a high biodiversity because nothing's really been interrupted. It's if something goes out of balance and Azotobacter is that a good example? Yeah, that'll do. It's a nitrogen-fixing non-symbiotic bacteria. So if that's not in function, that means that there's enough nitrogen in the soil here, but it'll have its spores sitting there. So once it goes out of balance, it'll get into its favorable conditions. The moisture will come up. It'll see that its electron acceptors and donors are all that sort of thing happening, and it'll start in its growth and development. And then that's the advantage of having the biodiversity. We're having things that are sitting there that can take over when they need to be taken over. Because how has Earth survived right up until 200 years ago? Quite well, hasn't it? Green, thriving. Man's come in and does his bit, and oh yeah, you should do stubble burning. Oh, stubble burning reduces the soil of 8 to 10% of the sulfur content. And isn't it interesting that quite a few charts that I've seen in Australia has that exact amount reduced in their wood. Anyway, focus on soil microbiomes is back to the bugs. Back to the microbes. They're not bugs, they're smaller than bugs. I should see, I get a bit, this is my topic. Of all my studies, plant, soil science, microbiology stuff, I really like agricultural microbes. I know a fair bit about them now, and I'm getting them to work with me for me. So if I get flustered and stuff, just bear with me if you can, because I'm just a bit excited because I really like this topic. I'm just going to stop it, see if there's anything being said or anything. Like 420 Fred, g'day 420 Fred. All right, back to it. Oh, and I'll just add too, there's a really cool study that's related on this topic. There's 3 people, I suppose I'll say at the end as well. I'll try to. I'm sharing my screen. Yep. This one, The Importance of Microbes for Sustainable Agriculture. It's written in November 2022. Oh, that's when it's published. So it's quite recent is what I was sort of getting at. But it goes through a chart in here that I'll go up examples of plant growth promoting microbes, and some of their types, their species, their strain number, which is the most important, and what they do. And people say, well, what's the strain number? Why? Well, because if I go and get Acinetobacter RG30, I'll expect this. But if I just get Acinetobacter RSC9, I'd expect it to do these. So from the studies that people have done, they've gone and put microbes under the blast system. Anyway, they documented of their strain name because they do specific things. Another example, Azosporium. No, it's a different species. But yeah, anyway, that's back to that. And then this, I'll go through that later. This is, so I'll put this in the description below for the DOI and the link for you to peruse this later if you want. Back to today. The soil microbiome. Well, the soil consists of a complex ecosystem of microbes known as the microbiome. They interact with each other and all sorts of things happen. The interactions influence soil properties, the soil quality, the depth, the structure and texture. So this picture here shows the different types of microbes that they are and how they work within each other. They're symbiosim networks, interactive networks, and interkingdom interactions. So they just have everything's linked. The biosphere. What do you call that? Oh, there's a sweet slide. The soil role of soil microbes. Decomposition, nutrient cycling, soil aggregate and structure, disease suppression, salinity regulation, symbiotic relationships, and phytohormone production. So if you can get the microbes only do usually a few things, so you need a balance of them. So that's why having a biodiversity and a large amount of them gives you a good balance of all these things here. Actually, I'm not going to go into my studies. Let's just get to these. What this says: microbial groups in the soils. So there's a few different soil microbe groups like bacteria, actinomyces, fungi, soil algae, protozoa, and bacteriophages, and they all do different things. Well, bacteria and fungi, you know, are quite different. So they're different cells to start with, but each one of those has their genre and species that also do different things, and they get broken down. So by knowing the groups, you can generally, that's where it starts. I'm not going to read out all of them because they're all broad. But I've got to remember too, people aren't up on my level. Microbial groups in the soils, bacteria. The most dominant group in soil microbes, involved in organic matter decomposition, biogeochemical cycling, and antibiotic production. We'll see, that's why I didn't want to read it, because there's a lot more stuff than that they do. That's very generalizing, so I'm not going to go into it. Microbial interactions are so important because by people thinking just by whacking microbes in, everything's going to be sweet, just put them in. What happens when you put in heaps of humans together? They fight. And some get on, some don't. Some need each other because some are disabled. Some interact successfully, some don't. So that's what this is. Some are leeches and parasites, as you know, some humans are. So by getting the right interactions, it's mega important. So by having a soil microbiome already established, it's already done this for you. So by disrupting it, you can see how important it is to keep it established once you get it there, and to get the right microbes cycling for you. So the different types of reactions: symbiosis, so mutualism, commensalism, which means A benefits off B. Harmful interactions, agonistic type things like amelitism, where A harms B, or parasitism, where A harms B, but B gets benefits off A. Actually, I think there's a few more than that. I remember a few more in some other microbe courses, and maybe they've summarised them back to 4 now. Symbiosis, commence. Well, what's the other ones? I think I should just go through this. can't remember, so I'm not going to. Microbes enhance soil through various mechanisms supporting its overall vitality, recycling of waste and detoxification, growth medium for plants, raw materials, help maintain soil moisture, carbon storage, nutrient cycling, control of pests, weeds, and pathogens, and create soil structure. Very, very important. This is all what microbes in soil fertility and to do in crop production. All these things. So if you were to grow in inert media, you would expect a reduced amount of all of this. And that's why you'd have to bottle feed it, give it pesticides. So this is the organic approach. This is a really good understanding of making microbes work with your organics. Because if you're with the normal pesticide type crew, you will put pesticides out and all of these microbes you've gone and built up will be destroyed in one instant. It's I don't know. I don't want to get in there. So this slide says plant growth promoting rhizobacteria, PGPR. So they're in the rhizobacteria that are in the roots. So PGPR enhance plant production, particularly in pathogen-free conditions. Well, they might, because they've got a free roll of what to do, but in a good soil, healthy soil, you're supposed to have 50% pathogens, 50% normal things, microbes. So I should just be reading this. See, I can't help myself. In my studies now, I've got a good overall understanding from the quite a few microbe courses I've done. Probably nearly done more microbe than blooming plant courses. So back to this. PGPRs act as eco-friendly biocontrol agents by competing for resources, excluding pathogens by producing antifungal metabolites like hydrogen cyanide, etc. PGPRs also induce systemic resistance in plants, like through the jasmonic, what's that, the was an exam question, that one. Anyway, through jasmonic and ethylene signaling, blistering plant defenses. It's key bacterial components such as lipopolysaccharides, flagella, cytophores, and volatiles play a crucial role in induced systemic resistance, promoting plant health and resilience. And on the left here, this slide shows by direct PGR mechanisms, through all the different ways that they help. And the indirect mechanisms, like through this induced systemic resistance or the prevention of plant diseases, is a way that they also help. And down the bottom, the PGPRs help by the cytophyll production competition and just being there, so that allows the pathogens to keep at ease and not proliferate. Microbial phytohormones. Plant physiological functions like growth, development, and responses to environment stimuli are regulated by various phytohormones. These hormones interact cooperatively or antagonistically. Plant growth promoting bacteria can synthesize or degrade certain phytohormones and produce volatile organic compounds, which enhance plant growth and influence phytohormone expression. They do a lot more than that for you too, if you get the right microbe working for you. So here's some examples of what they can do. So Azospirillum, Brassiaceae, and Bacillus produce cytokinins, which promote cell division and overall plant growth, especially in shoots and roots. Rhizobacteria, like Azospirillum and Bacillus, produce indole citric acid, which helps in roots, improves the plant's ability to absorb water and nutrients. Bacillus ligniformis, Bacillus pumilus, I use both of them, produce gibberellins, which help cell elongation, making the plant grow taller and develop more branches. And also, gibberellins they swell fruit. So here's the classical plant phytohormone, a bacterial substance based on their plant growth. So plant phytohormones plant growth promoters and growth inhibitors. So they're the phytohormones that happen in plants. They all do. Wow, they put ACC dismutase in there. What's that? I didn't really see that last time. Okay. And the growth inhibitors. Well, they can be, we go into why they do. That's what happens. Plant substances affecting the hormones, regulation of the plants, growth promoters, and growth inhibitors. The role of the plant microbiome phytohormones in different plant growth activities. So they all have an effect. Actually, no, they don't. Because some are good for some things and some are bad for the others. Like ethylene, it says before was bad. Well, it's good for germination. It promotes germination for the radicle to come out of the seed. But after that, it's not good for growth and development. And then it goes into the ethylene into flowering even. Produces the femaleness in flowers. So they all have different things that happen. So it nearly looks like the Mulder's antagonistic nutrient cycling chart. Should see if there's anything written. Genetic Memory Farm says, good evening, peeps. Hope you're safe and happy. Good day, Genetic Memory Farms. Microbial phytohormones enhance plant stress tolerance through various mechanisms. There's the DOI up the top right if you want to read it. But it's to plants. There's abiotic and biotic stresses. I'm not going to read out different types of them. Abiotic means non-living things. Biotic means living things. So the abiotic and biotic stresses. There's heaps of them, as you can see on that slide. This is a chart that I found years ago. Frontiers in Microbial. It's very important. It gives you the microbe and the phytohormone that's produced from it. And you can use this to your advantage. I've used it a lot. Righto, section 1, section 2 now. Climate resilience. So rising global temperature and increased extreme weather events have driven climate change, make adaptation crucial to protect plants and ecological systems. So agriculture is already affected by climate change, which disrupts plant homeostasis and causes stress from factors like drought, salinity, heat, and flooding. Microbes assist plant in managing abiotic stress like drought and extreme temperatures. My throat hurts. Shit. I need someone here to talk off. You talk now. So these are some processes here. Jeez, my throat hurts. Oh no. Climate change, yeah, well, that sort of just explains what I said there in an easier way to read it. You see the different osmotic stresses to do with water and oxidative stresses to do with the oxygen. I'll find a lolly or something. Hang on for a sec. Let's see if I can get something to chew on. Mouth dry. Is it moist? So there are numerous mechanisms that help in promoting growth in stress conditions induced by climate change. Two of them are reduction of ethylene level and countering the effects of high salinity in plants. So Pseudomonas species support plant growth under stress by producing hormones, emitting beneficial compounds, reinforcing root barriers, reducing oxidative stress, and various other things. These actions mitigate stress and enhance growth, ultimately improving yields despite the adverse conditions. That's one example. So, I'm trying. Gosh, there's always something in it. Love to read all these. That's the big bit. Decreased oxidative stress. Because plants get stressed like we do. So if you can allow them improve. Damn it. See if there's any questions. Nothing. Half an hour, eh? Wrong one. What if I stop there? How far do we get in? Just into it. What about into bioremediation? We get up to that. Righto. Reduction of ethylene levels in plants. So Pseudomonas strains promote growth under stress by producing ACC. Okay, I'm not going to go into the technical stuff. Ethylene regulation, mutual benefits. So if you can get them to reduce the ethylene production, it will not have the effects of the bad growth and development, countering the effects of high salinity in plants. Okay, so this is Pseudomonas mandeli, and the strain there improved salt tolerance in wheat and pea by enhancing suberin and lignin decomposition in the xylem cell walls. So that's an example of finding the right one, the right strain to work with you. So microbes are just brilliant. And this is just one actual species of microbe that's doing this one thing. So if you can get a few thousand different types, you can see what's the effects are going to be. I'm working with a dozen, and just a few I've put in do the job that they should be doing, and I'm getting the results that I should be getting. This is all really, really good stuff. So that's just showing the cell pathways of how it's traveling. Right, I don't have to go into those details. I think you really want to know that. Bioremediation. That's where I was going to stop. Because there's a few things in bioremediation. My voice was hurting. All right. If you want to, the importance of microbes, I might stop on bioremediation. It's an easy way to stop. It's nearly halfway. The importance of microbes for sustainable agriculture. It's another study that you can read in your spare time if you want. Here's the DOI, I'll put it in the description below. It's got some good stuff. It's written in 2022, so it's up to date. So that's all I want to put in. Actually kidding, I'm not even sharing. So that last few minutes, I didn't even share about Pseudomonas. And I stopped there and then went back to this one. We're talking about ethylene, and that's countering the effects of salinity. An example was Pseudomonas mandeli, the strain number IBKI 14. Sorry about that. Thrown off with my voice. It's just a little bit better now from that lull. I've got to maybe do that next week. Fair income. All over the shop. So excited for today too. Oh, I'll stop on bioremediation because you need good substrate and grounds that you're working with, fields everywhere. People say, I've heard, should be talking about it next week. I wonder if I should continue. Been going half an hour. My voice is a smidgen better with that lolly. There's one person watching. Thank you for that one person. I can stop. I want to make it interesting. Yeah, and I'll talk about next week why bioremediation is mega important, because people think I don't need that. Everywhere needs it. Even healthy places, they need healthy soil that's cycling very well, so you can do all sorts of positive things with bioremediation. So I'll talk about that next week then. You hear my voice going weird. I don't use my voice enough these days. That's probably why. Oops. Oh no, I'm still not sharing the screen. Unreal. All right, I'm going to end there. Thanks for watching. We're still into lecture 1, and hope you can join me next week. Do the same thing again. Microbes in agriculture, woohoo, university course. Put your comments in below. Press the like if you liked it, please. And I'll see you next week. Happy breeding, happy growing, good health to you all. Bye-bye.